So what we're going to do today is start with the fungus of the day, which is one of my other favorite fungi. Um, and then we're going to go through the Chytridium mycota. Okay, we're going to the four phylum we're going to go through: Chytridio, the zygo, the asco, and Basidium mycota. And we'll start with the chytrids. Now, when I talk about the chytrids and I talk about these different phyla, the phyla are identified and identifiable through their sexual structure primarily, okay? So we are going to go through some life cycles, and I'll point out what I want you to know in the life cycles and what is just not expected for the exams and what is expected. So don't get freaked out when I start talking about mitosis, meiosis, and ploidy. Those will be important, but I guarantee you you'll be able to catch on with what I'm trying to describe. All right. Before I start, are there any questions? And Nate, you're monitoring online stuff, I guess. Unless Tom's on there, he can monitor the chat. Any questions before? Any questions before we go to the exam? Because the exam will be like anything we spoke about that you didn't understand if you were reviewing. Okay, upstairs. All right. So the fungus of the day is the humble mycena. These are a really cool group of fungi. They're really small mushrooms, probably not taller than a couple of inches high. And you can find them all over the place, in almost every area on this planet. Not so much probably in the tundra, in the Arctic or Antarctic, but pretty much everywhere else you can find them. And they come in all kinds of cool colors. The ones you're going to see probably around in the woods in and around Ohio are going to be little brown ones or little yellow ones. And I'll tell you a little bit about the yellow ones in a couple slides. But they have an interesting history of where the name mycena came from. So you can go to the next slide and let me, rather than turning my head, I'll read it from up here. The name comes from an interesting origin. It basically comes from Greek mythology. A popular legend has it that Perseus founded Mycena. It was called that because he was thirsty and a mushroom sprang up, and he provided him with a stream of water. So what they did is he drank, and the nobles, where was it? He plucked the mushroom because he was thirsty. It was a poisonous one. Now, that's one thing. These mycena do not tend to be poisonous. So this theory or this myth from mythology may or may not be as accurate, but it was a small mushroom. He started hallucinating, seeing a city all around him. So the nobles, who were anxious to curry favor with the king, took all this very seriously and founded the city upon the spot where the picnic was. And so it was that they established a city based on eating this one mushroom. Now, the mushroom gave him, gave him visions and was said to be hallucinogenic. So he probably was eating psilocybe, which we'll have a whole lecture on hallucinogenic mushrooms. Actually, if you're interested, we have an entire course in our department called Psychedelic Studies that talks about them pretty in depth. But he was probably eating a psilocybe of some sort, not a mycena. But it's a myth of how this small group of small fungi actually got their name. Now, interesting at the bottom, which is kind of neat, that city later became a thriving center for illicit drugs. So maybe the hallucinogenic part was somewhat correct. Next slide. Okay. Oh, is that? Yep. So there's, I've just put this up there because there's a couple different stories of how mycena got its name. And here's just another one, again, talking about drinking and water springing forth. So this is kind of a neat thing to go along with this mushroom. Again, I always try to say, I try to link fungal biology and fungi and mycology to things that are not just in biology. So here's for those classic majors, little something. Next slide. So if you look at the mycena, there's many different types, and they range from kind of whitish, like I said, most of what you're going to see is brown, but this one is a real cool one because you can find this all over Ohio. This is the walnut mycena, okay? it only grows on the husks of walnuts. So it doesn't grow on wood. It doesn't just kind of pop up out of the soil. It only grows on the husk of a walnut. So if you see this little yellow mushroom, again, only about an inch or two high, and you look around and you see a bunch of walnuts on the ground, or you're near a walnut tree, you can be darn sure that those things are growing on a walnut. And it's kind of a fun little trick. What you can do is get like a knife or a shovel, whatever, and just dig down a little bit into the soil, and I guarantee you'll find a walnut. So even if you don't see a walnut, but you see that little yellow, kind of yellowish orange mushroom, you're definitely going to find a walnut under it. Kind of good, fun trick to show your friends and family when you're walking through the woods and you see the little guy. Usually if you see one of these little yellow ones, you're going to see dozens of them. They are kind of small, so they're not going to pop out when you're just walking around. You're not going to see them, that quickly. But if you do notice you're around a walnut tree, it's worth kind of crawling around and looking for them. And again, these, like all mycenas, are edible, but it says inconsequentially edible. Basically, most mushrooms you find in the woods, just about most of them, you can eat. But this falls in the category of why would you eat it? It doesn't have much flavor to it. Maybe you can give a little of that umami mushroom scent to food, but not really. So, but if you're hungry and you're desperate in the woods, yeah, go ahead and eat it. I call it intestinal roulette. You may not die from some of the mushrooms in the woods you eat, but also it might give you an upset stomach or gastrointestinal problems. So any questions? Next slide, I think that's the last one. Oh, on my mycenas. No, there's this one. This one's really cool. You can find this one in Ohio too. This is the bleeding mycena. And it's called bleeding mycena because you see a picture of someone's fingers there and they squeezed it and this red pigment comes out and it looks like you cut yourself. And it's really interesting because mushrooms have been used by many different cultures. Amy's going to talk about this a little bit either Wednesday and Friday. But mushrooms were used by many cultures as a dye, and they have a number of different pigments in them. So this one has this beautiful red pigment. You can see it up there. It looks like blood. It actually will stain your fingers for a little bit. Doesn't wash off too easily. But it's another one that's kind of fun. If you're walking in the woods, you see these little brown things. Squeeze them. If it's red, it's a bleeding mycena. Kind of neat. All right, Nate. Okay. Any questions about the mycena? Again, just trying to show you some things that you might see in and around Ohio. Okay. So we're going to talk about the Chytridium mycota. And we talked about some of the general features already. But what I'd like to spend the rest of today's lecture before the quiz going over is one that's been very important internationally as a pathogen of amphibians. By pathogen, again, it's growing on living tissue, and it's going to be doing something to the detriment of whatever it's growing on. If you have a problem, if you have feel queasy when you see pictures of dead or dying animals, I'm going to show some pictures of some sick frogs on here. You're welcome to put your head down or whatever. I just giving you a fair warning. Just these pictures, and it's not going to be the last time you're going to see pictures of animals or insects and such that might make you feel a little sick. Okay, let's see some dead frogs. Okay, and I'm going to use the podium here so I can use the cursor a little bit. Okay, so this is chytridiomycosis of amphibians. Mycosis is causing disease. And what we have here are A, B, and C is a healthy frog. D, E, and F is an unhealthy frog. So the fungus, again, this is a chytrid. So chytrids have what kind of spores? You say it loud. Moving spores, which are also called come on, guys, you got a quiz today.
Zoospores?
There you go, zoospores. And the zoospores swim because they have what? What allows them to swim? A flagellum, okay? So with the chytrids, Most of them will have one flagella, and they can swim. Again, they're not swimming far, not swimming across a pool, swimming very short distances, and those spores really don't last that long. But some of them have more than one flagella, but this one's going to have one flagella on it. So if you think about it, chytrids are mainly found in and around water. Well, so are frogs and newts and salamanders and other amphibians. So you can imagine they're both going to be in the same place, at the same time. So we talked about those zoospores, and they tend to swim towards something, okay, trophic. And I gave you an example. We can get some water from the Olentangy here and put your fingernails, some hair follicles, or spider webs or something like that, and you can get zoospores swimming towards it. So for this one, this chytrid likes keratinized material. And in a frog, the keratin resides in the mouth parts of a tadpole or the pads and underbelly of the mature frog. So that's where the fungus is going to be attracted to. So up here, if you look at A, you see a frog that's sitting flat and upright, sitting flat like a normal wild type would. In the bottom, the legs are kind of splayed out. In E, you can see the underside of an infected frog, and you see where that keratinized material is going to be. It's around its legs and its pad underneath. All right. So interesting. If you took a frog, you see in B that they're actually holding the frog down to look at its underside. Why is that? It can hop away, and they have a very strong righting mechanism. They flop back over really quickly. Good call. The bottom one, they're not holding it down. It's pretty diagnostic of chytridiomycosis on the frogs, is that it doesn't have that righting reflex. So if you put it up on its back, even if you don't see that red infected material, it's not going to flop back over. So it's relatively easy diagnostics. In C and F, it's basically, you can't see it so well in here. Maybe you can on your computers. It's a microscopic image of the dermal tissue, the external tissue of the frog. Interestingly, there's a student at Ohio State, not from my department, but another department. I was on her PhD committee that went down to South America and spent 2 years almost living in the bush, living out in the forest, tracking frogs and monitoring rivers and streams for the chytrid. So she'd collect water, she'd send that to a lab that she'd go visit every now and then down outside the woods, and she'd also collect frogs and see if they didn't have it or they had it, and then go back and recollect the same frog the next year or next season to see when it might have, if it caught the infection or not. Here's a neat question. How do you think she could tell if she caught the same frog more than once? They didn't tag them. Some of them are really small, so they're a little difficult to tag, so that's what you would think. They actually cut their toes off in different patterns, so you know which frog is which. Seems kind of barbaric, but they'll nip, one toe off or maybe two, and make a list of which frogs have what pattern on them. The other thing is, with the frogs, how do you think, well, I'm not sure if I get to this in the next slide, but I'll just say it anyway. Frogs, when they mate in the spring, okay, they mate in these big piles, okay? Big piles of many different frogs. So it seems like a nice place to spread it frog to frog. We really aren't that sure if that's the main way it gets spread or if this gets spread from contaminated water that they happen to be splashing around in when they're a tadpole and then once they become more mature. But yeah, those mating piles is one hypothesis of how it spreads so much. All right, next slide, thanks. Okay, so this is a life cycle. This life cycle is really a simple one. When we get to the Zygomycota, Asco, and Basidiomycota, I have it's going to be much more complex type of life cycle. So this one's pretty easy. And when I would talk about life cycles, I always like to start in a similar place when I discuss them. So we're actually going to start here at the bottom, okay? I always like to start with the sexual or the asexual spores. So in the bottom, we have these zoospores that come out of a zoosporangium. We'll talk about that in a minute. They swim in water and find their host. So they're going to swim again towards keratinized material. In this instance, it's a pretty frog. I don't know the name of it. Harlequin frog. I don't know. So what's going to happen is the spores are going to swim. It's going to find a frog, and once it finds that frog, it actually attaches to and insists in the frog's epidermis tissue. So that's when it's going to do all its stuff, all its bad stuff. Once it insists in that frog, that's when the frog starts to lose viability. It loses its righting reflex and all that. So we start with the zoospores. They're swimming along. They find a frog. They're going to lose that flagellum, right? So they're going to be not motile anymore. They're going to insist, form this cyst on the frog, and that's going to be called the sporangium. It's a word that we're going to use over and over. You might see it on your exam. You won't see it on the quiz today. None of this is on the quiz today, by the way. But a sporangium is just basically, as it sounds, it's a case or a house where spores are formed. So the zoospores will insist, and the sporangium starts to develop. And inside that sporangium, you find all the zoospores. So it's a pretty simple life cycle. Zoospore forms a zoosporangium that grows and matures, and inside of it, more zoospores form. And it has this little cap, all right? This little cap, in other fungi we'll call it an ostial, but it's going to pop, and all those zoospores are going to come squirting out. Now, we saw this in the video I showed at the end of last lecture on Friday, where this little cap came off and all these spores came spewing out, swimming out. That's what's going to happen here. And then the whole thing happens all over again. Now, it's kind of interesting. This 23 degrees Celsius is a critical temperature. The research that the student did when I was on her committee kind of debunked that, but it's still believed it needs relatively cool temperatures for fungus to live. Once this poor frog gets infected, it's going to die in 2 weeks. It's kind of stinky. So next slide, let's learn a little bit more about fungus itself. So I made a point of saying that this has caused a lot of problems worldwide. And you can see every dot up there is a place, and this slide's rather a little bit dated, but it serves the same purpose. Every dot is somewhere that this has been found. So what's your first observation? Okay, so Northern Europe, China, India, you really don't see many dots. Why do you think that is? Well, right, places where it's moist. What else? Thanks for letting me know. Okay, places where there's active trade going on is an idea. Also, these probably didn't look terribly hard for them in those places at the time the slide was made. I'm sure if we updated, you'd find it now. Now, you see there's plenty in America, okay? We do find this chytrid plenty of places in the United States, in North America. Interestingly, though, our frogs tend to be resistant, okay? So most of the frogs that are dying are the small ones down in South America, not your big bullfrogs that you're going to find in and around here, or leopard frogs that you find in and around Ohio. You probably could find a leopard frog, swab it, and we could probably, using some DNA tests, find some of the chytrid on it. They're just not getting sick. So they do have some resistance in certain types of frogs. So keep this in mind that, we'll put this slide or talk about it again. But the question is, a couple that we're going to be thinking about in the next few slides. First of all, has this always been around? If it's been around for a long time, why is it all of a sudden we're getting extinctions of frogs down in South America and other places? If it hasn't been here for a long time, where did it come from? And we'll try to answer some of those questions a little later on. Or why is this happening now? And you can think of some ideas while we talk. Okay, next. Yeah. Oh, good question. No, that's the reaction of the frog to it. Yeah. That you don't really get to see the fungus. It's microscopic. You wouldn't be able to see it, but that's just the manifestation. So here's what's happening. So BD is what we're going to call it because it has this huge name. I call it chytridiomycosis because I'm not so good at pronouncing that big long name. But most people that study it call it BD. So how does it infect? Well, we just learned it in cysts and forms zoosporangium on a keratinized material. It disrupts the ion balance in these frogs. So when a frog's laying flat, like on a wet stone or something like that, it's actually absorbing and balancing its ions, specifically calcium. Calcium, you'll see, I didn't have it up here, but calcium, you'll see, has major effects in a lot of biology. They say, was it one calcium ion exists for 4.3 femtoseconds or something like that, free in a cell, even in your cells. So calcium, when you disrupt the calcium balance, you disrupt a lot of things. Specifically, when you disrupt calcium balances, you affect the ability for your muscles to work properly, which includes your heart. So these frogs, it messes up with the calcium pumps not working properly. It messes up with their ability to use their muscles and flip over, and also they eventually go and have a heart attack. So all these, I keep saying frogs, but all these amphibians are basically die of heart failure in the end. So there have been localized extinctions. Some areas have had some amphibians recover, and I don't think we really know how that has happened. Maybe there's a small population that have some natural resistance to this fungus. Maybe that spurred on some of this recovery. And like I said earlier, there's no apparent impact on the frogs in Ohio. Next. Okay. So and let's get some answers even from people online too. Why is this happening all of a sudden in the last decade or two? Why has this not been a problem, for a longer time? I mean, how these frogs even evolved if they're all dying of heart failure? Okay, so it could be tied to some sort of global warming, and maybe this is an ecological indicator that something's changed. Maybe that temperature variation, very fine change could cause this to become virulent or cause the frog to be more susceptible. Okay, so climate change is one we just suggested here. It could be it could be an invasive species, and by invasive species, it came from somewhere else. So it evolved one place and then was brought somewhere else. You can think like kudzu in the south. And there's a lot of others that we'll talk about. Emerald ash borer, it's invasive. Okay, kill all our ash trees. In your book, I can't remember if you've been through the chestnut blight chapter, again, invasive species. Okay, so those are two options. What else could be going on? So it came from somewhere else, climate change, there could be a bunch of others. Say it, Len? Okay, so maybe this was an isolated area that had this chytrid, and then with floods or, let's say, deforestation and erosion, it spread to a larger area. That's a very good option. Yep. Okay, so there might have been changes in the frog's environment. So their nutrition, or maybe there's some sort of pollution that's affected the frog's ability to clear these pathogens, where before they used to be able to do it. And that's an option too. So these are the things that some scientists here at Ohio State and scientists all over the world are trying to figure out what happened to make either the frogs more susceptible or make the pathogen more virulent. So it could have come from somewhere else if we think just about the pathogen side. Fungus could have mutated. They mutate all the time. And there's one mutation that allowed this to happen, and since it was successful, evolution, then the pathogen became more successful and made more pathogen, and that mutation allowed this to happen. There's ways to figure all this out. So how would you figure out if this pathogen's new or it came from somewhere else? Go to the next slide real quick, and might go back. Yep, thanks. Take a guess. How would you figure out if something's invasive and came from somewhere, or it's been there and evolving in that spot? Oh. Okay, so we can look at its DNA, and if we look at the DNA, what would we be looking for? Because you're not going to really be able to find that one mutation that easily, but what would you compare it to? I can't hear you. Oh, okay. It's a good answer. It's the right answer. But what would we compare? What would we do? Mm-hmm. So if there's a high diversity, okay, that means it's going to be evolving in that spot for over a long period of time because it's always changing. Every generation, some changes happen. If it's a low diversity, meaning all the DNA is the same, from all the isolates we isolate from a certain area or a certain country or a certain region or worldwide, if they're all the same, that suggests it came from somewhere recently because it hasn't had time to evolve and build up mutations in its genome. Yes? Yeah, well, both of those would require us to know a lot more about it at the beginning, but good answers. It's a way to study them. The DNA is the quick and easiest way to do it. And that's basically call everyone that's been isolating this fungus or go find a bunch of dead frogs, isolate the fungus, and then look at the DNA and look at the genetic diversity. So if we go to the next slide. I have it up at the top. It's getting cut off a little bit, but that's basically what scientists did. They looked for diversity, and they found that genetically, the fungus is identical in almost all the samples they took. I think I have no difference between collections from sick frogs or healthy frogs. So it's not like there's one strain that is super virulent and healthy frogs just have a different version of it. They found the same DNA sequences, the same genetics in fungi isolated from healthy frogs, dead frogs, sick frogs, and wherever else they looked. So what does that tell you? No genetic diversity suggests, it came from somewhere, right? One population came from somewhere and spread very rapidly across the planet, across the country, so it wasn't that if it was a single mutation in one area, you wouldn't see, first of all, this lack of genetic diversity. You also wouldn't see a rapid increase all across the world like we saw with this one. So it suggests that a single line was recently expanded globally. It could have been a version of the fungus that's typically a saprophyte, and it gained the ability to be a pathogen. We don't know. All we know is that it's one version that would spread around the world. We also do know that there are some frogs that are resistant. Like I said, there's no effect to the leopard frogs or bullfrogs you find in Ohio. So they could have been a reservoir, and then those frogs were sipped or sent somewhere else, or got out, got loose, maybe down in South America, and started spreading this guy around. Okay, go to the next slide. Okay, so let's pretend that it did come from one spot. How would it get spread so widely, so fast? And we're talking, this probably started happening in the 70s, 80s, 70s, which is still very recent. True. Now, this is in freshwater. It's not in ocean. Yeah. It's a good answer. It's a valid answer. It did come from freshwater. Doesn't really do, there's probably versions that grow in saltwater, but this one's a freshwater bug. How could it get spread so fast so far? Yeah. Say it again. Yep. I mean, they're pretty frogs, you know. And so one hypothesis is that it was shipped all over the world as pets or either illicit or legal animal trade. Because they're not found just on frogs. Right? Like I said, they're found on amphibians and other things, other amphibians as well. It's just the frogs seem to be the one that everyone's paying attention to and monitoring now. That's probably what happened. I don't think I have another picture of a slide, did I? I mean, another slide of the world. No, go back. No, that's fine. Yeah, go back to the world one. I thought I usually put one in. So if we came to the conclusion that it probably came from one source and got spread around, how do we try to track down where that original source was? Again, it comes down to genetics. You can track through genetics, you can try to find an area where it has this lineage, this virulent form, but also has a lot of diversity in and around it. So it would be in an area that has a lot of chytrids that have this variant, but also have a lot of other variants in and around, which meant it came from an area with a high genetic diversity. If you look at this map, you see a couple places that have a lot of pins in it versus others. One of the biggest and densest areas in southern Africa. I always like to joke around that in America, we never tend to take credit when we export problems, but we blame other countries when we get them. Well, we're going to blame Africa on this one. So the way the genetics work out, it probably came from southern Africa. There is a hypothesis that's pretty much been debunked, but it's kind of a fun story. You guys are a little young, but if someone said, back when I was a kid, to a woman, said, "Hey, did the rabbit die?" What would that mean? You ever hear it? You can ask your parents. They used to use rabbits for pregnancy tests, okay? But it was very invasive for the rabbit. They'd have to basically euthanize it and see if it was ovulating. What they do is take serum from a woman, inject it into the rabbit. Well, they found they could do this with frogs. And the frogs, first of all, you didn't have to kill the frog. Basically, if you took the serum, the hormones in the woman, a pregnant woman, would cause the frog to basically spew out eggs or start producing eggs. I forget the exact thing. So frogs are shipped all over the world for pregnancy tests at one point. So one hypothesis laid back, it's a fun story, is that the frogs are shipped around and carried this, and that's one way. And they didn't die when they did the test, so they kind of released them and they start hopping around, and next thing you know, you got problems on your hands. So that's kind of a fun story. Most likely it was shipped around part of a pet trade or something like that. And it was shipped to a lot of different places. So it was somewhere you could do a lot of international trading, and in southern Africa, the whole thing makes sense. So, Any questions about this chytrid? Now, this isn't the only chytrid. There's, what did we say, 100 and some that have been studied. This is just the one that's most important. There are a couple, like I said, that do infect plants, which, oh, sorry, which we're interested in, obviously, but there's not much other research on them. Okay. All right. Yeah. Oh, yeah. Did the rabbit die? When rabbits were used as pregnancy tests. So it's kind of a snide way of asking someone if they got pregnant, if they wanted to or not. Say, hey, did the rabbit die? I heard it when I was a kid, but it's an old term, old phrase. I don't think anyone in any year that I taught this class that anyone's kind of, maybe one person came up with the right answer, an older guy. Okay, so we have like a couple minutes before the quiz. Are there any questions before online or in person here about the quiz? It's 10 points. Nate and Tom will grade them. If you have problems with your grade, talk to them.
Where do we locate the quiz online?
How are we doing it online? All right.
Yeah,
So for those that notified us that they were going to be online, Nate and or Tom put their have your emails, and they're going to email you, send you, I guess, email you the quiz as soon as I pass them out in the class, and then you have till, I mean, 10 minutes. It's only going to take you a few minutes for the quiz, but then you're going to email them back to Nate or Tom, or I guess Nate, whoever sends it out. You? Yeah, send it to Nate. Or do you want them to put it in like Carmen box or something? Yeah, just it's whatever you guys want to do. Yes. Oh, ha. Actually, I didn't either. I'd use the board. That's okay. I get it. I should not assume that. As long as it works. All right. You guys take one aisle, the other takes the other aisle. It should be fine. The copier broke in our office this morning too. I had to use the scanner or the fax machine. Pen. Try to write legibly. Sometimes it's. And then, did you already send it to the online folk? Okay. Nope. You need a quiz? Or? Oh, okay. Sure. Yeah, just do the email. So again, if some people came a little late, if you're going to miss a quiz or an exam, let me know a couple days ahead of time. Makes it easier for us.
If someone online can receive the email, just let me know, and I'll send you the quiz they set.
Hmm? Do what you can. You don't have what? Oh, here. I'm sorry. I will bring pens next time, or forewarn you to bring some. Yeah, is it a fresh one? Brand new. Okay, so usually at this point, I let you guys take it from here. When you guys talk a little bit, speaker, I want you to listen. Put on the UV light, they're all stale. Because we keep these.